Hi, my name is Jordan, and my project that I've been working on is expression profiling of epithelial transcription factors in breast cancer cell lines. So, first, I'm going to give you a little bit of background. When you think about cancer spreading throughout the body, it is defined as cancer metastasis. And this is just the spread of cancerous cells from a primary location to other organs in the body. We know that 90% of human cancers are of epithelial origin. And so, in order for the cancer to be able to move, it must require invasive properties in order to metastasize. And normally, this happens during epithelial to mesenchymal transition, or EMT. So, here I have a figure of what EMT is. And so, during EMT, epithelial cells gain mesenchymal characteristics. Normally, epithelial cells are non-motile and they are normally adherent to each other through cell to cell adhesion. And mesenchymal cells are motile, and these allow for cells to gain access to the bloodstream and move throughout the body. So when you think about metastasis, it's normally when cancer cells obtain mesenchymal characteristics during EMT. And so you can see here in the figure that the white cells, which are right here, are normal epithelial cells, and the pink cells are tumor cells. So the tumor cells look very similar to the normal cells since both of them are epithelial. Then some of the pink cells undergo EMT, which you can see here at the end, and they start to be able to move and they become elongated. And that's when the cancer can then access the bloodstream and move throughout the body. We also know that transcription factors, which regulate the transcription of genes in EMT and as well as in MET, can uncover the mechanisms by which the processes are activated and controlled. And so, in order to test our hypothesis, we decided to profile their expression in a panel of epithelial and mesenchymal breast cancer cell lines using real-time PCR. And so, you can look at my figure at the top here, and PCR is basically just the amplification of DNA. And this method allowed us to measure relative gene expression and mRNA levels. So, for our results, we got, if you look here at figure 2, first, I'm going to explain how the graph is divided up. So the first blue line, we have the control, and then we have luminal cells, and then we have basal cell lines, and then we have mesenchymal cell lines on the very last part. So we found that the following transcription factors, OVO1 and 2, GRHL 1, 2, and 3, ELF 3 and 5, and IRF6 were expressed at high levels in the majority of epithelial cell lines. And that is both the basal and the luminal cell lines, because you can see here in the graph that the lines for the relative mRNA are very high on all the epithelial cell lines. And we also found that GATA3, FOXA1, and ESR1 had a high amount of relative mRNA expressed in luminal cell lines, which you can see here at the top. The luminal cell lines have a much higher relative mRNA compared to all the other cell lines. If you look at the second graph here, the white bars represent the epithelial cell line, and the black bars represent the mesenchymal cell line. And so we also saw the OVO 1 and 2, GRHL 1, 2, and 3, ELF 3 and 5, FOXA1, ESR1, and IRF6 showed a higher expression of relative mRNA for the epithelial cell line. So when you think about transcription factors, these can help regulate if something's going through MET or MET. And so there are specific transcription factors that induce both EMT and EMT. For example, OVO induces the mesenchymal to epithelial transcription in human cancer, and ZEB1 can repress OVO1 and OVO2 genes. So those just help regulate if it's going through MET or EMT. So in conclusion, our data suggests that the expression of the 11 transcription factors is associated with the epithelial phenotype and that they can potentially suppress EMT. We also found that the MET transcription factors are highly expressed in epithelial subpopulations, and they're defined by the CD24 positive and CD44 negative markers. And in the future, we plan to identify EMT suppressors, and this will allow us to facilitate efforts to inhibit EMT and metastasis in breast cancer. Thank you for watching my presentation.